Imagine a world where you can literally reassemble the building blocks of life inside a petri dish, mimicking the chaotic spark that started it all. That isn't science fiction anymore. It is happening right now in Zurich labs. In this video, we dive deep into how scientists are recreating the very first steps of evolution to create synthetic cells with their own metabolism and self-repairing abilities. We will uncover the secret experiments involving tiny microbes and RNA enzymes that might one day allow us to heal our bodies by rewriting biology itself. This is the moment where life meets technology, and it is going to change everything you know about what it means to be alive. Let's transport ourselves straight to the laboratory of Dr. Kassar at the Swiss Federal Institute of Technology in Zurich. Here, they aren't just studying cells, they are engineering them from scratch. Their recent breakthrough involves recreating a wild endosymbiotic relationship, a scenario where one organism lives inside another and both benefit. But this time, it is entirely synthetic. Biologists Gabriel Geiger and Julia Vorholt took a massive leap by simulating the exact conditions that led to early life on Earth. They managed to create a system where synthetic membranes, previously thought impossible to sustain, actually developed their own internal metabolism. This means the cell isn't just a static container. It is a functioning biological machine capable of processing energy and nutrients independently. By successfully replicating these ancient interactions in a controlled environment, they have essentially paused time and rewound biology to its earliest moments, proving that we can build life from the ground up with unprecedented precision. So, how did they turn raw materials into a living entity? The answer lies in the concept of putting life into the origins of life. As Dr. Kassar famously puts it, scientists have mastered the art of creating life's fundamental building blocks, but those components alone are merely inert parts waiting to be assembled. To bridge that gap between chemistry and biology, they had to apply lessons learned from evolution directly to the design process. Instead of relying on random mutations over millions of years, these researchers introduced specific catalytic RNA enzymes, or ribozymes, into their synthetic cell models. These ribozymes act as the digital code for biological function, triggering crucial steps that would have been vital during the origins of life. By carefully curating these components and embedding them within a self-sustaining membrane structure, they created a collection of RNA enzymes capable of catalyzing important metabolic reactions. This approach effectively shortcuts billions of years of evolutionary history, allowing us to engineer cells that behave with the complexity we usually reserve for natural organisms. Now, let's talk about what this means for our future, and perhaps a little bit of cautionary philosophy. When you can recreate a scene from billions of years ago in your own lab, you are no longer just an observer, you are the director. Dr. Kassar makes a compelling point that while we have become adept at constructing these building blocks, they do not yet exhibit behaviors we would classify as alive. There is a distinct line between a sophisticated chemical reaction and true living consciousness or sentience. The challenge now is to push past this threshold by applying evolutionary principles more aggressively. If we can successfully replicate the spark of life in a lab, we must also address the implications of playing God with such precision. This raises profound questions about identity and definition. If we can create synthetic cells that metabolize and adapt, do they possess any rights? Do they require protection? The science is advancing rapidly, but the ethical framework to keep pace with it is still being written by philosophers and scientists alike. We are standing on a precipice where our ability to build life will determine how we define humanity itself for centuries to come. But let's look at the practical side of this incredible achievement, because that is what truly excites us all. By mastering the ability to recreate early human development in a lab, we unlock the door to a future where regenerative medicine is not just a dream, but an achievable reality. Imagine a world where stem cells are no longer limited by their natural lifespan or the body's inability to repair itself. If we can harness this regenerative potential of synthetic cells, we could revolutionize how we heal, repair, and even rejuvenate the human body. Think about treating degenerative diseases like Alzheimer's or Parkinson's not just by slowing them down, but by actively rewriting the cellular machinery that causes them. We are looking at a future where the body is no longer a finite machine subject to decay, but a dynamic system capable of self-repair. The implications for biotech applications are staggering, offering us tools to fix errors in our DNA and restore lost functions with the precision of a surgeon and the versatility of nature. So there you have it. From Zurich labs to synthetic cells that breathe and metabolize, we are witnessing the birth of a new era in biology. We are learning how to put life into the origins of life by combining ancient evolutionary lessons with cutting-edge engineering. The potential to heal our bodies and rewrite life's code is right around the corner. If you found this deep dive into synthetic biology fascinating, please hit that subscribe button so you never miss an update on how science is reshaping our world. Drop a comment below telling us what you think. Should we be creating more of these synthetic cells? Let's start a conversation about the future of life itself.